Okay, hi everyone. I'm Lila, and today I'm going to talk to you a little bit about the work that I've been doing for my PhD project, and then I'm going to switch gears slightly and talk to you a little bit about my journey to learning about equity, diversity, and inclusion in science. Um, so to start with the flat oyster, so the European flat oyster is a species of oyster native to Europe that's been harvested for consumption for centuries. So Austria edulis actually translates to edible oyster, so they're supposed to be pretty tasty. So they've been economically important, but historically overexploited. They're also vulnerable to parasites such as the Bonamia parasite, and for this reason, we've been seeing these population declines. And there's interest in sort of coming up with some management strategies for this species. An important first step when we want to determine management strategies for a species is to disentangle the population's genetic structure. And so previous work by Lepeg and colleagues in 2020 determined that there were four major population genetic subgroups of flat oyster in Europe, which we can see with the colors here. And this was using a small number of genetic markers. So for my project, we used whole genome sequencing to sample four representative locations. So the ones in the circles over here, so that we could have millions of markers and take a closer look at the population genetic structure. So the first thing that we did was to determine if we were seeing the same results as with the smaller number of markers. I don't think this works. Oh, does that? Oh, yeah, it does. Okay, cool. So we can see that the populations separate out really nicely. And on the x-axis, we have this potential geographic effect where we're seeing the North Sea and the Black Sea on, the North Sea and the Atlantic on one side and the Black Sea on the other. And on the y-axis, we have this potential lineage effect occurring, where interestingly, we see that the North Sea and the Black Sea, which are the most geographically distant, are the closest together. So the thing about these overall sort of views of population structure, such as a principal component analysis, is that they're a very simplistic measure. And so it's important to sort of take a deeper look at what's going on. So a principal component analysis uses all the markers. So it's looking at a whole genome-wide pattern rather than at specific locations within the genome. It also neglects to take into consideration structural variants, such as chromosomal inversions, which may also have a different differentiation pattern. So chromosomal inversions are these sections of the chromosome that have inverted 180 degrees, and therefore the genetic sequence is no longer reading in the same direction. And so what this means is that recombination, or the exchange of genetic variants, can no longer occur at these inversion regions. And therefore, inversion regions may maintain different genetic variation patterns than the rest of the chromosome, even in the face of gene flow. And in populations where individuals have chromosomal inversions, we can see three types of inversion alleles. So we can see individuals that are homozygous with no inversion, individuals homozygous with the inversion allele, and individuals that are heterozygous, so they have one chromosome with the inversion allele and one without. So chromosomal inversions are really interesting because they can maintain this large section of advantageous allele combinations. And for that reason, they've been associated with different environmental ecotypes, such as in freshwater rainbow trout, forest and prairie deer mice, and sand dune sunflowers. And in the flat oyster, we see that we have these three chromosomal inversions popping out. So what you see here is a dot plot. So we're comparing the chromosomes of two different individuals, one of which that has the inversion allele, one of which that doesn't. And we can see that in chromosome 4, we have this little inversion at the bottom. So where we see the nice diagonal is where we've got the correct orientation. And then when we see the little missed diagonal, that's where we have the inversion. And then at the beginning of chromosome 5, we have a slightly larger chromosomal inversion. And then chromosome 9 has this huge chromosomal inversion, which is taking up about half the chromosome. And we can have a closer look at these inversions using a local principal component analysis. So what this is doing is it's taking a principal component analysis at different sections throughout the chromosome so we can see where different differentiation patterns pop out. So we've got the principal component 1 score on the y-axis, the position along the chromosome on the x-axis. And then we see these inversion regions popping out. So we see the 3 groups emerging that correspond to the groups that I showed you earlier. So we've got the individuals that are homozygous for one allele, the individuals that are heterozygous, and the individuals that are homozygous for the other allele. And now I'm going to tell you briefly about parallel evolution, which is the repeated independent evolution of similar phenotypes or genotypes by closely related lineages. And so, as we'd seen previously in the principal component analysis, we saw this sort of strange link between the North Sea and the Black Sea for the European flat oyster. But we also see this popping up when we look at the inversion allele frequencies. And parallel evolution is something we've seen a number of times in nature. So we see this in the 3-spined sticklebacks, which have repeatedly independently adapted to freshwater habitats with changes to their plate armor. And we've also seen this in the timamau walking sticks, which have adapted to different host plant species. And so inversions in parallel evolution end up representing this sort of gap in the literature where we have a lack of studies that are investigating the intersection between them, but we know through simulations the inversion should create a stronger signal of parallelism faster. And so we want to know what the relative contribution of inversions to parallel evolution is in nature. And so I decided to take a closer look at parallel evolution throughout the flat oyster genome using the F4 statistic, which for simplicity's sake is just a measure of parallelism. So if we have the F4 score on the y-axis, the position along the chromosome on the x-axis, we can plot all the different genetic markers. And if we see a score, a neutral score of 0, this suggests that there's no parallelism occurring. If we see a positive score, this suggests a parallel signal. A negative score indicates an anti-parallel signal. We can create thresholds for this and determine if there's markers that are showing a signal. So I did this across the whole genome, and interestingly, as we would expect, we see that these chromosomal inversion regions show a strong signal of parallelism. But what was most interesting was that we see that the strongest parallel signals actually occur throughout the genome at these so-called islands of parallelism. So we're seeing that parallelism is not just confined to these inversion regions. And in conclusion, we see that the flat oyster has 3 inversions present in different populations at different frequencies. There's a parallel signal between the North Sea and the Black Sea present throughout in these islands of parallelism. We need demographic history analyses to further untangle this fine-scale genetic structure, which is what I'm working on currently. And translocations and breeding programs need to consider this fine-scale structure when choosing the origin of stocks. So thank you to all of my team members for this work. And now I'm just going to switch gears and talk a little bit about my journey of learning about equity, diversity, and inclusion, or EDI, in science. So my journey began many years ago, 10 years ago, where here I was as a little baby EDI student when I was an undergraduate at Queen's University in Canada. I joined the Social Issues Commission and I was a member of CARED, which was the Committee Against Racial and Ethnic Discrimination at Queen's. And I learned a lot during this time. And so I was super excited to take this into the biology department when I joined as a master's student. So I was the first student representative on their newly established committee of EDI. However, this excitement soon became disappointment when I realized that, frustratingly, we talked about a lot of things but really didn't get much done, which I think a lot of us working in EDI can relate to. But in the end, it was actually my project itself, which I think taught me the most about inclusion in science. So I was working on the northern fulmar, which is a Canadian Arctic seabird. And this project was part of a larger interdisciplinary project concerning the future of Canada's seabirds. And so these seabirds inhabit breeding colonies that are situated on the traditional territories of Canada's Inuit people. And it's these communities that were really the driving force for this project, as they were the ones who provided most of the sampling effort and also held the knowledge of where these colonies were. And so during this project, I ended up learning a lot about traditional ecological knowledge. So one fun fact I love is that the historically largest population of northern fulmars, which has sadly experienced severe decline, is known as Kakudlui, which is the Inuktitut word for northern fulmar. So this shows us how this sort of ecological knowledge is woven into the language. So I learned a lot as well about two-eyed seeing, which is this idea of using both the lens of indigenous knowledge and Western science. And I had a lot of opportunities to attend workshops, including one at Arctic Net. And this got me thinking about this idea of the Arctic being this very sort of hot research topic that's very exciting to work on. There's lots of changes in the Arctic, but a lot of this research often neglects to consider the communities that are living there, that are most impacted by the changes, and really are the biggest stakeholders in the research we're doing there. And so overall, this brought me to really think about who our science is for, who benefits from our science, who's excluded from our science, what's driving us to do our science, and how does our science enforce colonial systems. So it really led me to realize that our science doesn't exist in a vacuum. And despite the fact that now I'm working on a very different study system, this is something that, like, I hope that we all can keep considering while we do our research. So thank you.
Thank you so much. So do you have any questions?
Hey, thank you. Like, it really resonates with me. I'm working in Australia partially, so like the topic you lastly talk about is quite resonating. Were you able to like share this thought with the people you were working on at that point during these conferences? Yeah, I'm just wondering, like, did you have a community of scientific with you talking about it and like reflecting, or were you just alone with these questions?
Well, so it was a little bit of a mix of both. I was very lucky in that the interdisciplinary project I worked on involved a lot of different stakeholders and gave us the opportunity to have meetings with Indigenous groups. And a couple of the scientists I worked with were super involved in Indigenous communities and had spent a lot of time investing time in building trust in these communities. So I learned a lot from them. I think where I felt somewhat alone at the beginning was realizing that our actual research lab that was collaborating with all these other scientists had never really considered acknowledging sort of the territories or the people involved in this research. And that was a question I sort of took to my supervisor, who was super open to it. And it ended up being a really great experience for our whole lab, where we got a lot of advice from the Indigenous Center on campus. We ended up having some workshops organized. And I mean, I certainly don't think we were doing things perfectly by any means. Canada has a long way to go in terms of reconciliation with Indigenous people. But I did feel like I ended up having some really great conversations. And I think that it was really nice how much my team all sort of got together for that. So there was like a lot of learning in that sense. So I definitely understand your concern when you're entering also a culture that's quite, like, I hadn't grown up in Canada, so my base knowledge of these things was low. But if you are able to find, there's often these sort of knowledge holders who are really willing to talk about their knowledge with you. I think that's a really nice way to sort of connect with that.
Any other question? I think it's just a remark. I think it's nice that it nicely ties back to the very first plenary session by Autumn about how you can also find knowledge outside of, I guess, academia and what one might call peripheral knowledge. Because I'm not an ecologist, but for those who know me, I work half with ecologists, and when I go to these conferences, I'm always a bit questioned by how some people do field work and seem to come back with this feeling that they know everything. And I feel like without people there, they would not have even survived.
Yeah, absolutely. Yeah.
Yeah, I guess it's not really a question, but I think it's nice to see how it connects.
Yeah, I think it's relevant to a lot of us for sure. Yeah.